Greetings to you all. Welcome to Medical Biochemistry video lecture series. In this video lecture, I will be teaching you all about beta oxidation, especially the different types of oxidation of fatty acids. The fatty acids are the acids which are hydrocarbons with COOH group in their structure, and they are generally represented as RCOOH. And these fatty acids are the dietary component which are oxidized in the cell to yield energy. The fatty acids or the fats do not give instant energy, that is the role of carbohydrates, but the fatty acids gives us stored energy. The fatty acids undergo oxidation to yield energy. Beta oxidation is the chief oxidation method through which the fatty acids are oxidized to yield energy. But besides beta oxidation, there are certain other oxidation as well which are used in different ways by our body, including alpha oxidation, omega oxidation, peroxisomal oxidation, etc. So we will be dealing all these oxidation types one by one. If we see into this diagram, this is the structure of a typical fatty acid. Here, one on the right side, we can see this carboxyl group, which is represented as COOH, that is carboxyl group, and numbering from this group towards the left side, we have got a hydrocarbon chain. The first carbon is represented as alpha carbon, the second carbon is represented as beta carbon, and the last carbon, which is the farthest from the COOH group, is represented as omega carbon. Now, if this bond, the first bond, which lies between the carboxylic group and the first C1, that is alpha carbon, if this bond is oxidized, that is called as alpha oxidation. So that means one COH will be released at one time, alpha oxidation. If the bond between the C1 and C2, that is the alpha and beta carbon, if this bond is broken or oxidized, then first two carbon, they are released. So that means two carbons are released, that becomes actually beta oxidation. It is the main source. So I am going to discuss the details of the beta oxidation first. Now the beta oxidation occurs in mitochondria, and it starts from the outer membrane and then it moves to the matrix of mitochondria. So the fatty acid that has to be oxidized through beta oxidation has to first enter into the mitochondria. So they have to cross the membrane, and this fatty acid first need to be activated so that they can enter into the mitochondrial matrix, and then these fatty acids in the mitochondrial matrix undergoes beta oxidation. So we will start with the activation and entry of fatty acids into the mitochondria. The first of all, we will deal with this concept because every time the fatty acids need to be activated. So in this diagram, we can see clearly the activation and entry of fatty acids into the mitochondria, how it happens. So there is a carnitine shuttle system which enable the fatty acids acyl coenzyme A to enter into the mitochondria. So in the center we can see this whole box like structure. This is the inner membrane of mitochondria, like this. So this is the matrix side and this is the out cytoplasmic side of the mitochondria. So this one is the, which I have shown it with red markers, this is the mitochondrial membrane. In the membrane there is an enzyme translocase which actually transfers the substances from one side of the mitochondria to another side of the mitochondria, that means from outside to inside. So any fatty acid which need to be activated, first has to be converted into coenzyme A. That means there is a molecule carnitine which receives acyl group from the acyl coenzyme A with the help of enzyme CPT1, and this enzyme CPT1 is also called as ALT1. So the carnitine is converted into acyl carnitine with the release of coenzyme A. This acyl carnitine readily enters into the mitochondria with the help of the translocase enzyme, and inside the mitochondria the reverse reaction occurs, that is catalyzed by CPT second. So in this reverse reaction inside the mitochondrial matrix, the acyl carnitine is converted back into carnitine, and this is accompanied by the same enzyme CPT, but it is second this time. The full form of CPT is carnitine palmitoyl transferase, and this enzyme is also called as AT1, that is carnitine acetyl transferase. Uh, 1 in place of 1 we can write second, then it will be the inside enzyme. It will become the enzyme which converts acyl carnitine into carnitine. And finally, the same enzyme translocase transfers the carnitine from matrix into the outer chamber, that is outside the mitochondria, that is the carnitine is again available. So this cycle starts from the carnitine, and then acyl carnitine enters, it becomes carnitine inside the mitochondria, and then the carnitine again regained outside for the next activation process. So this is the carnitine shuttle, but during this shuttle, the acyl coenzyme A, which is now outside, becomes inside part, and this acyl coenzyme A is actually the activated fatty acid which is now used in the beta oxidation process. And this is how by carnitine shuttle system the fatty acids are activated and then they take entry into the mitochondrial matrix so that they can undergo further beta oxidation. Now we also can remember one point over here, that this carnitine shuttle act as this carnitine shuttle act as rate limiting step, because uh, if any of these enzymes is not functioning properly, or they are being inhibited by some other sources, then the entry of fatty acid does not take place and the beta oxidation is blocked. We will discuss it later on. All right. So these are the four steps. There are four steps. In the first step, acyl carnitine is synthesized. In the second step, the acyl carnitine enters into the mitochondrial matrix. In the third step, the acyl carnitine change into carnitine, and in the fourth step, the carnitine goes back towards outer side of the mitochondria. Now these four steps I have discussed here with the write-up. So this is all about the write-up. You can halt your video here at this point and you can read these points which I have discussed. So there are four steps for the activation and entry of the fatty acids inside the mitochondria, so that the further beta oxidation process can be carried out. Now it is a rate limiting step, carnitine mediated entry of fatty acid. Rate limiting step is also called as. Now when we go for oxidation, there are four important types of oxidation. I will start with beta oxidation. It occurs in the mitochondrial matrix, and in beta oxidation, whatever long the hydrocarbon chain is, in one cycle 2 carbons are eliminated out. So for example, if the chain is 16 carbon long, like say for example I have shown it here, 16 carbon long chain, then 7 cycles are required, and then last there will be 2 carbon molecule left. All right. So the number of turns depends on the number of carbon in the fatty acid, and in each turn there will be 2 carbon molecule eliminated out. So this cycle is called as beta oxidation because the chain breaks occurs first of all at alpha carbon and beta carbon, carbon number 2 and carbon number 3, and that's why it is called as beta oxidation rather than alpha oxidation. All right. So in case of palmitoyl acetyl coenzyme A with carbon number 16, I am showing you first beta oxidation starts from this place, and then second, third, fourth, fifth, sixth, seventh, like this. So as a rule, number of carbon atoms decides the number of cycles. Now here I have shown you the exact cycle. This is the beta oxidation cycle here, and these are the structures which are from where I am starting. So if we say for example, if carbon 16 chain long chain is there, then it is palmitoyl coenzyme A, and then with the help of enzyme acyl coenzyme A dehydrogenase, uh, hydrogen is removed, and then FAD receives that hydrogen change into FADH2. So palmitoyl will become, this is 16, now it becomes 14, that is beta unsaturated fatty acid in all coenzyme A. This beta unsaturated fatty acid change into beta hydroxy acyl coenzyme A with the help of enoyl coenzyme A hydratase. This step is called as hydration. The first step was actually oxidation of FAD. The second step is hydration. Now third step is again oxidation of NAD here. In the third step, the beta hydroxy acyl coenzyme A get converted into beta keto acyl coenzyme A. And again there is oxidation of NAD, which converts from NAD to NADH2. Enzyme is beta hydroxy acyl coenzyme A dehydrogenase. This is the enzyme. Now, in the fourth step, which is the thiolysis or cleavage, this beta keto acyl coenzyme A splits into these two components with the help of acyl coenzyme A acetyl transferase, that is also called as thiolase, into myristoyl coenzyme A and the 2 carbon acetyl coenzyme A. All at each step, one molecule of acyl coenzyme A is liberated out. So these are the steps. These are the 4 steps of one cycle. So say for example, if we have got here, we have got 16 carbon palmitoyl coenzyme A. So these are the acyl coenzyme A, acyl coenzyme A in first cycle, second cycle, sixth cycle, in the seventh cycle. In the seventh cycle, the last acyl coenzyme A will be liberated out. Now, each step we have seen that FAD is been formed, FADH2, NADH2 been formed, so that means 7 cycles. If there is any, then 7 molecules of FADH2 and 7 molecules of NADH2 will be formed, which will later give rise to ATP. All right. So this is the cycle of beta oxidation. So we have to mug up all these steps. No other way to remember this. We have to mug it up, and I have taken it from Lanenzer, the book of Lanenzer. This beta oxidation cycle is taken from there. So if we talk of the energy yield in beta oxidation, say for example, the example which I have taken is for palmitic acid or palmitoyl with carbon 16. The 7 cycles will be there for elimination of all uh, 16 carbon, 2 at one cycle. So 7 FADH2 will be formed, 7 NADH2 will be formed. So one FADH2 give rise to 1.5 ATP, that means 7 will give 10.5. 7 NADH2 will give 7 multiplied by 2.5, that is 7.5. And then 80 ATP are released by this step from acyl coenzyme A. Finally, जो acyl coenzyme 8 acyl coenzymes A are formed. So 8 multiplied by 10, because in one cycle 10 ATP will be formed, that through ATS 80 are formed here. So if it sum up all these, it becomes 108, and 2 ATP